Hello everyone. So in this question, we have a graph which is related to some plasma glucose concentration, as you can see here. Let's read the system first. When the blood enters the typical mammalian kidney, some of the plasma passes through tiny pores into the nephron, leaving behind the blood the rest of the plasma blood cells. Thus, the concentration of a small molecule is newly formed plasma filtrate in the kidney nephron is close to what it was in blood plasma. So this line is very important. If you look at the concentration of a small molecule in the newly formed plasma filtrate in the kidney nephron is close to what it was in blood plasma, means the blood plasma value and the blood concentration in filtrate, that can be same as well. Okay, so as this filtrate passes down the nephron's tube, some substances are absorbed from the filtrate, passing back into the blood, and other substances are secreted into the filtrate from the blood. These substances move across the wall of the nephron. By the time a filtrate reaches the bladder, its composition is markedly different from the plasma. So here we have x-axis in which plasma glucose concentration is given, as you can see, and the y-axis in which we have the glucose concentration in filtrate or urine. So this axis is a urine one, and this axis is my filtrate. Clear? So let's do some questions. So question number 1, at plasma glucose concentration of 6 mg per ml, the concentration of glucose in newly formed filtrate is approximately. So if you look at this line, it's saying the concentration of a small molecule in newly formed plasma filtrate in the kidney nephron is close to what it was in blood plasma. It means if blood plasma is 6, so you can see if blood plasma is 6, then filtrate will also be the 6. The filtrate value will also be the 6, this. And it is given in the stem as well, so no need to see the graph in this, especially in this question. It's very clear. So filtrate value and the plasma glucose value will be the same, so it should be 6. Now the next question, from the graph, glucose first appear in the urine at a plasma glucose concentration closest to. So as you can see, first urine starts with the 0, and then the glucose concentration in urine, which is 2 mg per ml, which appears at 4. When the plasma glucose concentration is 4, then the glucose in urine appears. It means this, at this point it starts increasing, and that is called renal threshold. Okay, and at this time, when the plasma glucose value is 4 mg per ml, then glucose concentration in urine is 2 mg per ml. Clear? So 2 mg per ml when plasma glucose concentration is 4. So glucose appears in urine at this value. So for this it should be 2 mg. Sorry, the question is talking about glucose first appear in the urine at plasma, so answer should be 4, because plasma is 4 here. Okay, so it should be 4. This is 2, the glucose concentration is 2 when the plasma is 4, so answer is 4 mg per ml, because it is talking about plasma glucose value, not the glucose concentration. So it should be 4. Next question, at 12 mg per ml plasma glucose, the filtrate concentration 12 mg per ml. We all knew filtrate value is equal to the plasma glucose, but urine contains only 8 mg per ml. How much glucose is being reabsorbed per ml of filtrate? So as you can see, it's saying the glucose plasma, the value at 12, then glucose value at 12 mg per ml plasma glucose, the filtrate contains 12. So this is my 12, and the plasma glucose is here 12, and glucose is also 12. You can see this one is a glucose concentration at 12. Clear? but urine at it, urine at it. So you know that glucose in urine appears when plasma glucose is 4 mg per ml. So from this data, the glucose concentration appears in urine. So it means we can say the reabsorbed value will be the difference of filtrate and urine. So urine value is 8, filtrate value is 12. You can see this is 12 and urine at 8. Clear? So the difference would be what? 12 minus 8, which should be 4. Clear? So for this question we can say, we directly say reabsorbed value will be glucose filtrate value minus urine. So this value is 12 and urine is 8, so answer should be 4 mg per ml. Clear? This energy gap is called reabsorbed value, okay? Now the next question, if plasma glucose is 7 mg per ml, how much glucose will appear in urine? If plasma glucose value is 7 mg per ml, let me remove some of the part. So it's saying 7 mg per ml. So this is the 7 mg per ml. And what should be the glucose concentration in urine? So it will be somewhere around 5. Clear? And you know that the urine glucose appears in urine at 4 mg per ml. And at that time glucose concentration is 2 mg per ml. Clear? This. So as you can see, this difference, because it's talking about how much glucose in urine, you can see glucose will appear in urine. It means we have to figure out the difference of 5 and 2. So you know that 5 minus 2 is what? 3 mg per ml. Clear? Makes sense? So this difference is 3 mg per ml. So this one is from graph interpretation. So thank you so much for watching this.